This is part 2 of the genetics lectures. Okay, so in the first lecture, you learned about a dominant allele. You'll remember the purple allele in the pea plants. And then there was a recessive allele. That would have been the white allele or little p. And you have a description here about what it means to be dominant and what it means to be recessive. And I also explained that in lab. I'll probably do it again next week when we get back into lectures. You also learned that a genotype is literally what type of genes do you carry, otherwise known as your genetic makeup. For example, you could be big P, and we said that that would make you purple as a flower. So if you're purple, that's your phenotype, that's your physical appearance. In other words, your genotype leads to your phenotype. If you were little p, that's a genotype, and you would be white as a flower. So that would be an example also of your genotype leading to your phenotype. You could also be big P, little p. That's also a genotype, and it could make you purple. That's another way to be purple. That's your phenotype. You need to know that genotypes lead to your phenotype. The type of genes you carry lead to what your physical appearance will be. And look at that. There's two ways to be purple. You could be purple with big P, or you could be purple with big P, little p. All right, we're going to have to add some more vocabulary to tell those apart. You also learned in meiosis and in the first lecture that the law of segregation is very important to genetics or your understanding of genetics. If you have, for example, big P, you're not going to give both of those alleles, both of those genes. You're not going to give those to one child. You're going to give them one or the other. Give them that one or that one. If you're big P, little p, you'll give them that one or that one. If you're little p, you'll give them that one or that one. You'll never give them both. Remember, if you give both, it's like giving two chromosomes. It's like giving both chromosomes, and that's the makings of a syndrome. All right, so no sooner does Gregor Mendel comes up with all this good information on pea plants and makes predictions about how they will look after crossing them. A short while after Gregor Mendel dies, the field of genetics is already up and running. Enter Reginald Punnett, the man who we give credit for the Punnett square. So the Punnett square is really useful to showing genetics crosses. And what we're going to do next is we're going to show you 4 basic genetics crosses. All right? And we're going to give this the title of Mendelian genetics. And they're called Mendelian genetics because there's a clear dominant, a clear recessive allele, and we can predict the type of genes or the genotypes and the type of phenotypes that will come out of this. All right, so let's do these crosses. The first cross we will do will be a cross between, say, a plant that has purple alleles and another plant that has purple alleles. All right, maybe we should go back and make a distinction. Do you see that an individual could be purple with big P, or purple with big P, little p? We're going to have to differentiate between those two. This one right here, we're going to call homozygous. And homozygous means that you have 2 of the same alleles. Or 2 of the same genes, big P and big P. Notice that this one has little p, right? This one is also going to be described as homozygous. Okay, but now we have another problem. We have this homozygous and we have this homozygous, but they look different. So in this case, we're going to say homozygous dominant And over here, we're going to say homozygous recessive. And that should make sense. We have the capitalized P's, the lowercase p's. This one down here is going to be heterozygous. That means two different alleles or two different genes, right? You have a big P, and that's different from a little p. All right, so now we need to get those terms down, and they're not terrible, right? If you have two of the same letters, you're homozygous. Example, big T, big T. You should have thought that's homozygous. Those are two of the same. They're both capital. And then you should have thought, and they appear to be dominant because they're capital, so that must be homozygous dominant, and you'd be right. What about little r? Well, that's also homozygous because it's two of the exact same letters, but these are lowercase, so they're recessive. That's homozygous recessive. And what about big S, little s? That's a heterozygous. And heterozygotes, they're just called heterozygous. You don't say heterozygous dominant or heterozygous recessive. That doesn't make sense. We just say heterozygous. Okay, so you learned some new vocabulary. In our first Mendelian genetics cross, we're going to cross a homozygous with another homozygous. And that could be the following. Let's just go with like earlobes, where earlobes come in two forms, free versus attached. The free earlobe is dominant over the attached earlobe. It goes like this. There's a gene for earlobes. And it comes with two alleles, a big F or a little f. If you get at least one big F, you're going to end up with free earlobes. If you get two little f's instead, you're going to end up with attached earlobes. Okay, let's be clear here. Every individual should have two letters, one from mom, one from dad. 1, 2. What I'm saying is that it doesn't matter if you have a big F, or a big F with a small f, you're going to end up with free earlobes because this guy already determined that. So it doesn't matter if you're big F, or big F, little f. You're going to have free earlobes. Just like we learned that as long as you have one capital P, you're purple. As long as you have one capital P, you're purple. It doesn't matter if you have big P, or big P with a little p. It doesn't matter. As long as you have one big P, you're going to be purple. It's the same idea. Okay, so if we crossed a homozygous, that is big F, crossed with big F, that's homozygous crossed with homozygous. And in both cases, they're dominant. You could also have homozygous crossed with homozygous. You could also have homozygous crossed with homozygous. All of those are the same thing, homozygous crossed with homozygous. And there are some differences. Homozygous dominant crossed with homozygous dominant, homozygous recessive crossed with homozygous recessive, or homozygous dominant crossed with homozygous recessive. In all cases, it's homozygous with homozygous. Let's take this cross and let's do it. We'll make it nice and big. Big F, small f. Doesn't matter where you put the F's. Could I have put the little F's on the side and the big F's up top? Sure. It doesn't matter. You're going to get the same answers. Okay, that's the outcome. Let's now look inside of that outcome to see what we got. First, let's list all the genotypes. What do we see? Let's begin here, and then let's work our way around. And I see big F, little f, and when I count, I see that there's 4 of those. And when I ask, is there anything else? The answer is no, there's nothing else. Well, this is a proportion now. There's 4 big F, little f's, and then there's nothing else. That's like saying that for every 1 big F, little f, There's nothing else. If you went into a classroom and you saw 4 male students and 0 female students, you would say for 1 male student, there is 0 females. That's a proportion, that's a ratio. So if we were to keep track of genotype ratio, or ratios or proportions, we would say that in this case it's 1 to 0. You can say 4 to 0, but that's redundant. 4 to 0 can be simplified as just 1 to 0. Also, if we go in there and after discovering the genotypes, we then ask, okay, then what do they all physically look like? What are their phenotypes? We would start with 1 and then work our way around. And we would see that it has at least one big F, and that means that you're going to end up being free. And notice that they all have at least one big F. So how many have free earlobes? How many have attached? There are 4 with free earlobes, 0 that are attached. That's the same as saying 1 to 0. And that becomes your phenotype ratio. It is 1 to 0. All right, you can go back and review that one. Let's do another cross. Cross number 2. This time let's do a homozygous, dominant, and let's cross it with a heterozygous. All right, what's a homozygous dominant versus hetero look like with earlobes, right? Remember again, free, that's having at least one big F, and it doesn't matter what goes there. Attached, you have to have little f, little f. Okay, so let's do this cross. This one would be big F, big F. That's homozygous and dominant. Cross with hetero. Let's do a big Punnett square. You can put the big F's on the side or you can put them up top, doesn't matter. And then you can ask, what are my genotypes? What are my phenotypes? In other words, what is your genotype ratio? And what is your phenotype ratio? All right, so for genotypes. For genotypes, you would notice that in the first box, you have big F, big F. And if you count to see how many there are, you would see that there's 2. And if you look over to the right side, you will see that there's also big F, little f, and that there's 2 of those. If you walked into a classroom and you saw 2 males and 2 females in the classroom, you would say that that's a ratio of 1 to 1. That's simplified. So your genotype ratio when you cross a homozygous dominant with a heterozygous is 1 to 1. And then if you ask, what do the phenotypes look like? You would see that they all have at least one big F. So there's only 2 possible physical appearances. You can have free earlobes or you can have attached. Free, how many are there? 2. Attached, how many are there? 2. A 2 to 2 ratio is the same as saying 1 to 1. So your phenotype ratio is 1 to 1. All right, you can review that one again if you'd like. Let's go to the next one. Cross number 3. This is a homozygous, recessive this time, cross with hetero. So it would be small f, big F, little f. That's what we're crossing. Again, I'll remind you that if you have at least one big F, doesn't matter what else you have, you're going to have free earlobes. And if you have little f, you're going to have attached. Let's do the cross. All right, let's take a look at what we got, what our genotypes are. Okay, start in one box and then work your way around. what do you see? I see a big F with a small f. How many of those are there? Looks like there's 2. And I also see little f, looks like there's 2. So that's a 1-to-1 ratio. And then I can look for phenotypes. Right? Remember, for phenotypes, it's what do you look like? Do you have free earlobes or do you have attached? And if I look in there, I can see that this one has at least 1 big F, and so does this one. That's 2. And I can see that these don't have any big Fs, so there's 2 of those. And that becomes your ratio, 2 to 2, which is the same as saying 1 to 1. So what's my genotype ratio in summary? 1 to 1. What's my phenotype ratio in summary? 1 to 1. Okay, you can review that one. Let's do one last one. Number 4. This is hetero crossed with hetero. What would that look like? That's big F, small f, big F, small f. Let's do that Punnett square, nice and big. Big F, small f, big F, small f, and then you fill them in. And then ask, okay, what are my genotypes? All right, start with one box, work your way around. I see big F, and if I scan around, it's only one of those. If I go to the next one, I see big F, and if I scan around, I see that there are 2. And then finally, I see little f, there's only 1. So my genotype ratio is 1 to 2 to 1. See, there's no math figuring out ratios. You just report what you see. So genotype ratio is 1 to 2 to 1. All right, what about your phenotype ratio? For your phenotype ratio, you're just, again, looking for the look. So how many are free? How many are attached? All right, so let's see. If I go in there and I look for free, I see 1 free, 1 free, 1 free. So there's 3 that have at least 1 big F, and there's only 1 that does not. That's a 3 to 1 ratio. So my phenotype ratio is 3 to 1. Okay, again, what makes this Mendelian genetics is that one gene has 2 alleles. One of them is dominant, one of them is recessive. And as a result, there's 4 basic crosses that you can do, and that's it. That's it. So the first thing you need to know are Mendel's 4 basic crosses. Let's review them really quick before we stop this video. In the first cross, you're crossing a homozygous with a homozygous, and you should know what those mean. That means having 2 of the same letters. And you should know that you can be homozygous dominant, cross with homozygous dominant. You can be homozygous recessive, cross with homozygous recessive. Or you could be homozygous dominant, cross with a homozygous recessive. In all cases, it's homozygous with homozygous. And if you do that cross, you're going to get an expected 1 to 0 ratio for both your gene types and for your physical appearance. If you cross a homozygous dominant with a heterozygous, you're going to get expected genotypes of 1 to 1 and phenotypes. Actually, this should have been 1 to 0. It looks like I made a mistake. Did y'all catch it? Oof. The phenotypes are all the same. They were all free earlobes. Glad I caught that. I'm glad you caught it too. 4 to 0 is the same as saying 1 to 0. Oof. Okay. Hope you caught that mistake. I'll have to put it in the notes on the video so that people know about it. Or maybe I should just leave it out and see if you catch it on your own. Okay, and then the third one, you have homozygous recessive cross with hetero. And that's going to give you expected genotype and phenotype ratios of 1 to 1. And then the last one, hetero with hetero, you'll get expected genotype ratio of 1 to 2 to 1 and phenotype ratio of 3 to 1. Okay, practice those 4 basic crosses. Try not to make mistakes. Oof, it's been a long day. Okay, and then meet me for the third lecture.